I'm Melissa Southey. I'm the Director of Research for the MPCCC. I'd like to open today's seminar by acknowledging the traditional owners of the land on which we are gathered today and acknowledge their elders past, present and emerging, as well as any members of the Aboriginal or Torres Strait Islanders community gathered with us this afternoon. Really a great pleasure today to introduce Ron Firestein to launch the 2022 seminar program for the MPCCC. Ron is well known to many of us, but in case you missed anything, he's head of the Centre for Cancer Research at the Hudson Institute. He's a physician scientist, both an anatomical and molecular pathologist. Specialising in cancer research, translational medicine, drug discovery and biomarker development. His focus is really on identifying new cancer targets and developing therapies for solid tumours. He's an established patent holder across a number of different topics and an extensive publisher in really high impact journals. But perhaps the most recent highlight, Ron, has been your capacity and great leadership to launch the Victorian Paediatric Cancer Centre, which Greg Hunt formally launched very recently from the Hudson, which was a terrific thing for cancer research across the state and I think across the nation. And I think with those amazing global connections, congratulations to your team and to the team in Parkville as well. So Ron, I might just hand it straight to you. I hope you can share slides with us. And Ron is going to discuss functional precision oncology, moving beyond genomic sequencing, which is an amazing title, very close to my heart, and I know close to many of our hearts. So Ron, apologies for the short introduction and the technical issues, but I hope we can hand over to you now for a flawless presentation. Thank you.
Thank you. Yeah, thanks a lot, Melissa, and we'll go ahead and get started. So the talk today, I'll be sharing with you, time permitting, two stories, really focusing on some of the efforts here in the Hudson Institute Cancer Center on functional methods to aid in precision medicine. So as we all know, the promise of precision medicine is really that we can target the specific mutations and alterations that are present in the cancer cell. And you can see that most of those targets really take the form in the variation in DNA sequence. And with the ever decreasing cost of sequencing, we're really now able to fastly detect mutations in virtually every cancer patient, and importantly, at a low cost. And that's really armed us with information regarding which patients may harbor a particular mutation and which mutation then may predict the response to a targeted therapy. And I'm sure here today we're all familiar with that concept. But at the same time, we really have to recognize that the fact that in the majority of cancers, those types of actionable genetic mutations are far and few between, particularly in childhood cancers, which I'll talk about, which is a relatively mutationally quiet type of category of cancers. Moreover, not all patients with these actionable mutations respond to the therapy. So still, even though the response is quite good, it's definitely not 100%, and that really underscores the fact that we need to understand the biology better. Moreover, the actionable mutations are not always tumor agnostic. In adult cancers, a really good example of that is BRAF mutations present in both melanomas and in colon cancers, but BRAF inhibitors really work well for BRAF mutant melanomas, but not so well for BRAF mutant colon cancer. So definitely we need to understand, again, the genetic and the tumor specific context in which these mutations arise and exist. And of course, importantly, targeted therapies eventually, as we find out, particularly targeted therapies for signaling pathways, are overcome in many cases by the cancer through a slew of different resistance mechanisms. So all of this really put together, I think, highlights the need for functional methods to identify therapies that go beyond the mantra of a single mutation to a single therapeutic response. And I will just, I think the timing for this talk is actually quite good in the sense that if you will look at the recent issue of Cancer Discovery, there are 2 papers and an associated review article by Tony Lethality, which really highlights new methods or approaches to integrate functional pipelines into the clinical setting, into a molecular tumor board setting. And this is just a schematic from one of those papers where essentially what they did is they layered in a drug sensitivity testing platform to go along with all the mutational and RNA sequencing analyses. And this was done for AML patients. But remarkably, they identified over 30 or 40% of patients were able to show sustained and effective responses to therapies that were not predicted by these sequencing platforms. And importantly, going beyond the N of 1 example, these type of responses can then be can lead to translational research to better understand the biomarkers of response so that we can then go beyond these personalized example to a more broader precision approach. So some of the 2 stories that I'll present today really kind of highlight some of our work in this area. So the first one is our work in the pediatric cancer space. And as Melissa alluded to, we've been working in this area for a few years now. I think one of the achievements we've recently had is really bringing everyone together under a single umbrella called the Victorian Paediatric Cancer Consortium, which is a really consortium that comprises the 8 major research institutes, hospitals, and universities, Melbourne and Monash University, under a single umbrella with the mission to conduct both clinical as well as basic research efforts to train the next generation of childhood cancer researchers and clinicians, and obviously improve outcomes for this malignancy. So these programs really span across the patient's journey from discovery research through clinical innovation, and a number of them really deal directly with precision cancer. Our program shown here in under the discovery research, I'll talk more about this, is called Next Generation Precision Oncology. And really the focus again is to go beyond the mutations and identify functional dependencies and biomarkers of response that are not necessarily driven by single mutation to single response. Additional programs are directed at particular tumor types, such as immunotherapy and tumor microenvironment, epigenomics and differentiation. Again, another precision medicine program led by Dr. Jason Kane with collaborators at MCH and RCH is around risk stratification, so using DNA methylation arrays to improve the diagnostic and predictive capabilities for sarcomas. And a larger program at Royal Children's Hospital around pharmaco and radiogenomics. So again, using genomic sequencing on each patient's germline to identify markers that may be associated with adverse response to chemotherapy. And around all these activities, we have common frameworks and resources such as consortium management, pediatric biobanking across both hospitals, data management and bioinformatics, and capacity building in the form of scholarships and other training opportunities. So I'd like to talk a little bit more in detail about the Next Generation Precision Oncology program. And that's really something that we've been interested at the Hudson for a number of years now, probably 4 years is when we got started with I think really the recognition that in order to advance new targets for childhood cancers, one needs to have good models to study those targets and progress them through the preclinical pipeline. And this is an area that's a real gap. As you can see here in these tables where we've shown what currently exists for adult cancer and pediatric cancer, we've definitely in pediatric, across the different pediatric cancers, there's been really good efforts and an understanding of the genomic landscapes of pediatric cancer, and integrating that knowledge into the clinic is ongoing. But where we're really lacking is models of the disease. So while adult cancers, we have literally thousands of cancer cell lines now, and some of you may be familiar with the larger scale initiatives coming out of the Broad Institute and the Sanger, where all these cell lines have really now been sequenced and screened across different platforms. But in pediatric cancers, there's really kind of a much smaller number of lines. Some of the types of tumors are not represented by any models at all. And in terms of functional annotation, it's really just beginning. So we saw this area here, particularly in model development and characterization of models, as a real gap where we could contribute. So with that in mind, we set up a program that really is not in one individual person's lab. So this is a program where a number of research groups at the Hudson have contributed. So my own lab, and then Dan Goff and Jason Kane's lab, teaming up with our clinical colleagues, both at Monash and Royal Children's Hospital. And we have a number of postdoctoral research fellows, bioinformaticians, and trainees working across this area. The real approach that we wanted to undertake is to use the data that's already available from the cancer genomic atlases, PET-C Bioportal, the Children's Brain Tumor Network, St. Jude's, and others, and begin to develop models that recapitulate the disease, and then use those models to begin to perform loss of function screens, gain of function screens, to identify new targets for pediatric cancer. So we started really locally at Monash and Royal Children's Hospitals, and recruiting about 40 to 50 patients a year through to our laboratory. That essentially involves taking the tumor tissue, dissociating it, and generating a model that recapitulates each patient. And then these models are really renewable resources. They can be cryopreserved. If the patient relapses, we can either generate a new model or get the original model out and work with the clinicians almost in real time to see which drugs might be working on the tumor from that individual patient. But what we really quickly realized is, because childhood cancer is a relatively rare disease compared to adult, even locally, we couldn't really get enough samples across all the different types of childhood cancers to begin to power and represent all the different tumors involved. So this is just a pipeline slide to show what we're actually doing with these tumors models. And you can see that involves molecular profiling. So we conduct whole genome sequencing, RNA sequencing, epigenomic profiling in the form of a TAC-seq and DNA methylations array, and together with Roger Daly's group, total and phosphoproteomic analyses. And then we've also generated Cas9 lines for each of our cell line patient models, and that undergoes CRISPR screening using a library that we've developed in-house. It's about 350 genes targeting oncology drug target agents, as well as parallel drug screens with about 2,500 compounds. I think it's important to put this into the landscape of childhood cancer precision medicine in Australia. Those of you in the space would have obviously heard of the Zero Childhood Cancer Program. So this program is really a landmark program in Australia to sequence and characterize childhood cancer patients at a national scale. And where we really fit in is generating these patient models and working with the Zero program to analyze them. And we've worked with Zero to analyze a cohort of about 15 models from this program. And these are some of the activities that are being undertaken. And it is important to recognize, as I mentioned earlier, that childhood cancer in more so than adult cancer really requires international collaborations. And that's because it is a relatively rare disease. Some types of childhood cancers, really there would only be maybe 50 cases a year in the whole world, and potentially maybe one or two or none in Australia. So in order to model those types of cancers, we really need to reach out to our colleagues. And I think this is one of the most exciting aspects of our programs, is that we've essentially over the last 4 years built these fantastic collaborations with both research institutes as well as clinical trial groups across the world. We have over 40 collaborators that have really heavily contributed and have been fantastically generous in working with us to build this larger scale childhood cancer atlas. So we have over 350 cell lines that we brought in. We characterize these lines, maybe only about half of them or a little more than half of them are lines which either recapitulate the tumor or are able to be grown in the laboratory. So one of the things that where we've given, I would say, back to the community is really being able to tell other researchers which lines are actually good resources and available for use. But I think what we're really interested is obviously, as I mentioned earlier, in using these cell lines to locally fine-tune treatment. And that's something we do on a case-by-case basis as the clinicians, pediatric oncologists may come to us and ask us to look at particular drugs or drug combinations. Obviously, the program is very interested in repurposing drugs that can be quickly repurposed from one indication to another. And moving forward, our probably biggest effort is really to use these tools for drug discovery and target identification. I'll also mention that we've built this data resource that we've now put online. So anyone interested in childhood cancer can actually access our dashboard of cell lines that are available. And this is the website, vicpcc.org.au/dashboard. And essentially, this will show you the active cell lines, the cell lines that can be grown, and the different modalities that we use and the number of cell lines that have been screened or analyzed by these different modalities. And this will form the basis of a data resource paper that will come out. And then later on this year, we're hoping to layer onto this an actual data portal where the data can be essentially interrogated and analyzed. So it'll have fill-in keywords that one can search for a particular drug or a particular gene and look for relationships between genotype to phenotype. So one of the first things that we did at the analytical level across all these cell lines is start to use multi-omic feature analysis, looking at the interplay between DNA methylation, RNA sequencing, whole genome sequencing, and other modalities in terms of being able to cluster these lines. And what we found is really the main features that cluster these lines are a combination of DNA methylation and RNA sequencing. And this is just the MOFA plot showing that, in fact, these lines really cluster very well with each other according to tumor type. So you can see the ATRT and MRTs cluster. Similarly, the adult high-grade gliomas cluster quite differently from the pediatric high-grade gliomas, which I think really, again, underscores the genetic differences between these histologically similar tumor, but genetically quite distinct. And as I mentioned earlier, some of the lines that we received, initially the researcher thought these are tumor lines, but we were able to actually show that a number of cell lines are actually not tumor, but they're tumor-associated fibroblasts or normal cells. And you can see those cluster quite distinctly as well. So really the power of this is that we can now, with these kinds of numbers, is build a cohort of childhood cancers that really span the different molecularly classified brain cancers. And childhood brain cancers is really one area where initially thought to be maybe one disease, but now we know that actually there are dozens of different molecularly classified subtypes of childhood cancers. And you can see that with our current cohort, this is the childhood glioma cohort. It's about 60 or so DIPGs, DMGs, and another 20 or so hemispheric gliomas. And you can see these cluster according to the main subtypes that are formed by the histone mutations, receptor tyrosine kinase activation, or MYC and amplification. And I think one of the exciting things now, we can begin to ask what are the differences generally between pediatric gliomas and adult gliomas. We can ask what are the differences between the diffuse midline gliomas and the hemispheric gliomas, and even now get into the molecular genetic subtypes, asking what are the differences between the H3.1 and the 3.3s, or any of these other co-occurring mutations. So this really gives us the depth to conduct that kind of analysis. So I'll just give you a few examples of how we're using this kind of data to make causal inferences and relationships between genotype and phenotype. And this is just an overall slide showing the overall data that we've gotten from about 445 drug screens performed across a cohort of about 150 cell lines. So you can see overall the number of hits across pediatric cell lines is quite similar to the number of hits in adults. And I think that just speaks to the fact that these lines, these cancers can be targeted by different agents. So it's not that we have less to work with than in the adult cancer space. But we wanted to go and now really identify targets and drugs that were specific to a certain subtype of cancers. And essentially what we wanted to weed out, obviously, is the drugs that have no effect at all, and that's shown here in the middle, or drugs that are effective but non-selective. So these are drugs that are essentially like bleach, they'll hit across all the cell lines. And you can see here in this kind of camel's hump type of pattern, the cell lines we're really interested in are the ones here which are selective. Essentially, across the whole cohort, maybe we don't get a high frequency of hits across the particular drug, but a fraction of the lines are sensitive. So we want to really understand what are the markers that denote this sensitivity. So this is just a plot looking at these potentially selective drugs. So these drugs here in the kind of the middle part of the plot are drugs that only hit in a fraction of cell lines. And I think one of the things that was really exciting to see is when we used machine-based learning approaches like random forest to essentially look for predictive features that predict the response, we saw that the majority of these different drugs could actually be predicted by a number of different features. So there are definite features that can predict these drugs, and that's the case across the majority of these drugs. So you can see here in the top part of the graph as opposed to the lower part of the graph. And this just illustrates two examples where we saw a single predictive features of dependency for a class of drugs. So in the first example, you can see that we have 4 different topoisomerase inhibitors in our drug library. And you can nicely see that P53 status, so wild type P53 cell lines are shown here in green. So these wild type P53 lines are predominantly the lines that are sensitive to this class of chemotherapy, the topo inhibitors. Similarly, and maybe not surprisingly, we saw that PDGFR-A mutation status is a good predictor of inhibitors across a number of different RTKs, such as the dasatinib that hit PDGFR-A, and you can see here in red, again, the mutant PDGFR-A mutant lines were exquisitely sensitive to those inhibitors. Similarly, across our CRISPR screens, we've performed 96 screens to date across a library of 350 genes. And you can see here in this heat map, in orange are the genes that are essentially what we call common essential. These are genes that, again, much like a bleach type effect, are essential for the growth of a cell line. And you can see that they hit nicely across all our cell line cohort, and they form essentially a positive control within the screen to tell us that these screens are working. In addition, we had other categories like oncology drug targets and other genes that are related to cancer, where you can see there were variable effects. Much like the drug screens, we saw again that the majority of our genetic effects could be predicted by different biomarkers. And these biomarkers really take the span of different modalities. So you can see here a few examples where in some cases, the genetic dependency was very much related to the tumor type. So ATRTs were highly dependent on PRC complex components, EZH2 and EED. Osteosarcomas were dependent on the ES1 transcription factors. And we can go beyond the tumor type and look at molecularly based classification. So for example, you can see that P53 wild type cells were exquisitely dependent on MDM2 and MDM4. And that's already been shown in adult cancers. But again, that shows us that this type of approach is working well in our pediatric cancer cohort as well. The next question we wanted to ask is whether we can go beyond, whether adult cancers and pediatric cancers, we had shown that we can see that they have both distinct as well as common genetic dependencies. But even for those genetic dependencies that may be the same, we wanted to ask whether the predictors of those dependencies may be different. And here, essentially, what you can see is a scatter plot showing the adult predictors in the x-axis compared to the pediatric predictors in the y-axis. Color-coded in purple are genes that are predictive, that have predictors of dependencies only in pediatrics. Similarly, in red are the ones that are only in the adult. And in orange here was with the class we're quite interested in are genes that are dependencies and have predictors both in adult and pediatrics. But the question is, what are those predictors? Are they really the same? And what we found is that in some cases, yes, they're the same. So for example, in MDM2, this is really related to P53 status, and that holds true for both pediatric and adult. And you can see here color-coded in green C are the common determinants of MDM2 dependencies, and you can see they're quite common between adult and pediatric. But conversely, we can see other dependencies such as FGFR1 dependency, where the predictors of FGFR1 dependency are completely different between pediatric and adult cancers. And that's quite important because it tells us that even if we can identify a drug that works, repurpose a drug from adult to pediatrics, the biomarkers or the markers that we might use to stratify patients in the clinic may actually be different. So it's something to keep in mind as we progress some of these therapies into the clinic. And indeed, just to show you a proof of concept, this is taken from Dr. Jason Kane's lab. He's used this concept of going from genetic analyses to drug screens to in vitro validation and then in vivo validation to a clinical trial quite rapidly. And this is really facilitated by this kind of streamlined approach where we can take the hits that come out of these genetic and drug screens. And in this example, we're looking at ATRTs, where he's found that HDAC inhibitors is a real therapeutic sensitivity in this tumor type, and has been able to progress this quite quickly and now move into a clinical trial called the North trial that's open for a number of tumors, including ATRT, looking at the effect of panobinostat, an HDAC inhibitor. So the last approach I'd like to cover is really the utilization of this kind of atlas for quickly sifting through genomic features. And as we know, when a patient's tumor or any genome is sequenced, there will be a lot of different variations found. Some of those are SNPs, some of those might be actually pathogenic mutations, but it's really about identifying some of these needles in the haystack. So across a cohort of different cancers, we might identify a number of different mutations, but in many cases, we don't know which of those mutations is pathogenic. So to take you through one example where we've used this integration of really the functional screens together with the omic analysis to quickly sift through, I'll show you just an example with PDGFRA. Again, using machine-based learning approaches, we've been able to prioritize particular mutations in a select number of genes as having a strong oncogenic effect. And using the CRISPR screens, you can see here that color-coded in red are potentially oncogenic mutations. Color-coded in blue are mutations in PDGFR that we are likely to be benign based on our predictive analysis. And you can see indeed that the CRISPR knockouts, that the majority of these lines with predicted oncogenic mutations are in fact hitting. In parallel, the drug screens that were performed completely independently largely align with this. So you can see the PDGFRA predicted mutation cell lines are exquisitely sensitive to dasatinib. We do see that 2 of the benign encoding mutants are also similarly sensitive to dasatinib. So that's obviously a discrepancy between the CRISPR screens, but we also recognize that dasatinib hits other targets besides PDGFRA. Again, that would be consistent with those being benign. So using this kind of approach, we can then layer on, and you can see here a mutation map, an oncoplot of the PDGFRA across the different predicted mutations we found. We've been largely able to, through single mutation knock-in or knockout analyses, recapitulate what our findings were from the larger computational analysis. And this really, I think, just points to the approach of using these large data sets to quickly be able to prioritize mutations into which ones may be likely pathogenic and which ones may be likely benign based on integrating these functional approaches. So just to summarize this part of the talk, we built a childhood cancer atlas that provides a repository of cell line models and molecular features, really representing a broad and deep collection of models of the deadliest childhood cancers. This enables streamlined approaches towards clinical translation and patient stratification. And we've also built this online database that can both provide the models and also the ability to analyze these samples by really anyone in the world with a computer and the internet. So what we've concluded thus far is, I think, 2 kind of broader conclusions comparing childhood with adult cancers, is that these pediatric cancers, they harbor both common as well as distinct functional drivers of growth compared with adult. And interestingly, even for common drivers of growth, we found that childhood cancers in some cases harbor distinct biomarkers of functional dependencies, really underscoring the fact that these are cancers that may harbor the same genetic dependencies, but the wiring of getting to that dependency may be actually quite unique. So where do we go from here? We are, as Melissa mentioned, we've recently received funding from the federal government in the form of an MRFF grant to progress our program. And some of the things that we're working towards are genome scale dependency screens of childhood brain cancer models, combinatorial screens using next generation CRISPR technology, together with Steffi Rosenbluth's lab, to be able to form unbiased combination genetic screens. And of course, we're very interested in how we can contribute in real time to molecular tumor board types of approaches for clinical impact, and hoping that not too far in the future that these types of functional approaches and drug screens can in fact be useful in a molecular tumor board setting. So we'll see how far we get with the second part of the talk. It's something a little bit more mechanistic, but really dealing with the concept again that in adult cancers, oh, sorry, my computer just get this back on the screen. Oh, doesn't look like. Some technical difficulties. Okay. So functional precision medicine for adult cancers. In this area, where we've really been focusing on is more in the discovery research and really applying synthetic lethality approaches to overcome cancer resistance. And I just, in the brief time, hopefully take you through one case example. So as many of you are aware, synthetic lethality is a concept originally derived from yeast that's been applied to cancer therapy, and quite successfully, in fact. Obviously, the clinical example of PARP inhibitors in BRCA mutant breast and ovarian cancers is a fantastic example of how this approach can lead to clinical impact. But really, the crux of synthetic lethality is that knocking out a particular gene A or gene B on its own may not impact cancer cell survival, but really through mutation or targeted inhibition of both gene A or gene B, one can then lead to a synergistic effect and cell death. So the importance of this pathway is really, of this concept is really been clearly understood, and there have been a number of large-scale efforts to explore synthetic lethality at high scale using genetic screens, such as the Cyclops screen performed at the Broad Institute, or combinatorial genetic screens and drug screens, and lastly, computational approaches looking for genetic mutations that can predict response across large databases. So where we've worked in this area is really across the MYC oncogene. So MYC is a well-recognized central effector of oncogenic transcription and transcriptional addiction in cancer. It's linked to a number of different important processes, including maintaining the stemness of cancer, translation, metabolism, splicing, and probably most importantly, promoting proliferation and inhibiting differentiation. So the second part of my lab is really working for over a decade to understand the different ways through which MYC is activated. And we've identified a number of both genetic targets as well as epigenetic mechanisms that are required to keep MYC transcription on. But in many cases, what we found is that the cancer cells can overcome these mechanisms when they are delivered on their own, and that leads to resistance. So to share with you one story, this is around are one of the targets, one of the genes that we work on in the laboratory called CDK or Mediator Kinase. Initially, we identified it through a number of integrative efforts through both RNAi screens, signaling pathway analyses, linking it to MYC and beta-catenin transcription, and showing that it was in fact amplified in a number of, in a fraction of about 25% of colon cancer cell lines. So CDK8 is a cyclin-dependent kinase. So as a CDK, it's obviously can be targeted by kinase inhibitors and part of this very large multimeric complex called Mediator. So we've shown that actually when you acutely inhibit CDK8, it has a very potent effect on cancer cells, both in vitro and in vivo as well. You can see that shRNA inhibition of CDK8 leads to tumor stasis or tumor inhibition with actually effects as well as on differentiation of the tumor. And importantly, one of the mediators of this is we've shown that CDK8 is important for MYC production and MYC transcription. And you can see that MYC levels are essentially almost abolished when you inhibit CDK8 in these tumor models. So what we've done to study CDK8 more closely is we've generated stable cell lines in colon cancer lines where we've knocked out CDK8 as well as its paralog CDK19, which can compensate for its loss. And to our surprise, we found that these cancer cells over many pathogens, they actually can grow completely unperturbed. So you can see two of these what we call double knockout clones grow exactly the same as wild type, despite the fact that MYC levels were essentially abolished in these cell lines. So this clearly suggested to us that these cell lines had learned to grow without MYC transcription. And that really led to a project by a talented postdoctoral research fellow in the lab, Danya Suraj, working with Claire Soon and bioinformatician to kind of look beyond CDK8 and identify what could be regulating transcription upon loss of this critical mediator kinase component. And this took the form of orthogonal, both high throughput drug as well as whole genome CRISPR screens that were conducted on wild type cells and cells that lacked CDK8 and 19. And what was really remarkable from these screens is the fact that you can see here from a volcano plot from the whole genome CRISPR screens. Maybe unsurprisingly, MYC was a high dependency in the wild type cells, but it was no longer a dependency in double knockout cells. So that's quite unusual because virtually all cancer cells depend on MYC, at least acutely. But what we found was a unique dependency is BRD2 and BRD4. So these are BET proteins, which are again, well studied and druggable. And you can see that they really came out specifically in our double knockout cells. And strikingly, they also came out as top hits in our high throughput drug screens. And you can see here on this scatter plot, distribution plot, that you can see the BET inhibitors. There were 5 BET inhibitors in the screen, and 3 out of the 5 BET inhibitors were really hitting across specifically in the double knockout cells. So what are these BET proteins? These are a cluster of bromodomain proteins. These are the phylogenetic tree of bromodomain proteins. There's about 50 of them. And you can see here 6 cluster within this BET protein family, and they're conserved in having 2 bromodomains, BD1 and BD2, and have already been shown through a number of studies in the past decade to be very important for transcription. And I think one of the things that excited us about both BET proteins and mediator kinase is that they're both druggable, and they both have drugs that are currently in clinical trials. And with CDK8 and 19, it's in phase 1, and BET proteins have been in a number of phase 1 and phase 2 trials. So we decided to validate these effects, and you can see across 5 different BET inhibitors, we found a striking reduction in growth, specifically in our double knockout cells lacking mediator kinase compared to the wild type cells. And again, you can see that more granular detail across these 12-point dose response curves in multiple cell lines. And importantly, we found that these effects were CDK8 specific, as overexpressing CDK8 at least partially restored the resistance to BET inhibitors compared to a kinase dead allele of CDK8 or CDK19, which was not able to do so, really underscoring the fact that these effects are kinase dependent. This was further validated in a number of genetically defined models, including mouse tumoroids. So these are intestinal tumoroids that lack APC. So we use isogenic tumoroids that either lacked APC or lacked APC in combination with knockout of the mediator kinase. And again, in this model, we found accentuated sensitivity to BET inhibitors. And that was shown also in xenograft models as well, where you can see that xenografts lacking CDK19 show exquisite sensitivity to the BET inhibitors. So lastly, to translate this closer to the clinic, we utilize the patient-derived organoid approach, and it's quite easy to generate organoids from colon cancers. So that's well described, and we generated a panel of different normal cancer and as well as metastatic cancer organoids. And just to summarize these results, we found that in fact, 4 out of the 6 patient-derived organoids, including from metastatic samples, showed distinct efficacy when BET inhibitors were used in conjunction with mediator kinase inhibitors. Now, we still have to understand why this is not occurring in all the patient samples. So the genetic biomarkers for the response still needs to be worked out, but quite encouraging to see that over 50% were responsive. Now, in order to understand the mechanism behind this synthetic lethality effect, we utilized both RNA-seq and ChIP-seq. And what we found, which was quite striking, is that when you perform bulk RNA-seq on these mediator kinase deleted lines, we saw a global reduction in transcription. So you can see here about 8,000 different transcripts. So about a third of the genome really is downregulated. And you can see these effects are non-redundant as CDK8 and 19 knockout on their own had very minor effects. So you can see CDK8 and 19 really have non-redundant or have redundant functions in transcriptional regulation. And this was complemented by a series of ChIP-seq assays. So you can see here ChIP-seq to RNA Pol2, which shows that in fact, there's a severe reduction in RNA Pol2 binding across both promoters and gene bodies. And that's across all promoters. So you can see this is really a global effect. And then we lastly asked what happens to BRD4 in these cells. And what we found, which was really, I think, providing a first explanation of what may be going on in these cells when you inhibit mediator kinase, is that number one, the BRD4, main mediator of BET protein activity, we saw increased overall binding at enhancer and super enhancer elements. But what was quite distinct was that most of the binding between BRD4 at these sites was co-bound with the MED12 protein, which is a part of the mediator complex. So we found that essentially the mediator complex still retains activity and is essentially compensated, at least partially, by the BRD4 BET protein. We lastly asked, what are the genes that are associated with these new sites that are bound by BET proteins? And what was really striking looking at some of these sites that are associated with these super enhancer or enhancer elements that occur in cells that lack mediator kinase, is that many of those genes are actually common essential genes, essentially genes that are required for the proliferation of any cell. And you can see here the plots from the dependency map showing that in many cases, these genes are just required for nearly 90% or more of the cell lines to grow. But what we found, which was quite remarkable, is that when you, looking at the expression of these genes in our different models, you can see really a stepwise reduction in the amount of these genes. So when you inhibit, when you take wild type cells and inhibit the BET proteins with JQ1, you can see a small amount of inhibition that occurs. Similarly, when you inhibit mediator kinase, you see a small level of inhibition, but really inhibiting both mediator kinase with JQ1 with the BET inhibitors together provides a much stronger level of inhibition of expression of these genes below 50%, likely explaining why these cells can tolerate inhibition of either of these transcriptional mediators, BET proteins or mediator kinase, but not both of them together. And that really led to a model that at least begins to explain some of these effects, where mediator is really known to be important for the transcription in cancer cells as well as in normal cells. But when you knock it out, the BET proteins essentially provide some level of transcription to maintain the growth of the cells, and that can be essentially knocked out by combining with BET inhibitors. So I'd like to thank our funding partners, both Children's Cancer, Robert Conrad Dawes, MyRoom, and other foundations, Victorian cancer agencies, and federal and state government that have provided funding. The pediatric cancer team here, Monash Children's, Royal Children's Hospital collaborators, the bowel cancer group, particularly Danya Suraj, who conducted the majority of the assays that I've described, and the CBTN group. And take any questions in the remaining 4 minutes.
Ron, thanks so much for an amazing tour around a phenomenal repertoire of functional assessment. Please just put your hand up if you have a question, or pop it in the chat. I can see both at the moment. But Ron, I wondered, just as a high level question. So, beyond genomic sequencing in clinics at the moment, I guess particularly relevant to the activities of the MPCCC, delivering genomic sequence from a tumour to clinical practice. So getting the sequence, interpreting the sequencing, delivering useful information in a clinically relevant time frame is still challenging. So to think about how your type of approach, which is so much more boutique, where do you see it's going to hit first? Where are you going to be able to deliver clinically relevant, timed information via these amazing approaches to characterization?
Yeah, that's a really good question, Melissa. So I think I pointed at the beginning to those 2 cancer discovery papers where they've actually been able to deliver real-time functional information. The caveat to those approaches is they were done on in the setting of AML, in hematological malignancies, where I think the cells are more homogeneous. It's easier to obviously take single cells and treat them with drugs. I think there's real challenges, obviously, with solid tumors in terms of, number one, growing enough cells, as well as differentiating the different heterogeneous cell types that come along with a biopsy or a resection material compared to what you can do with cells from the blood. So definitely major challenges there. And also success rates are not perfect. I think, there are different technologies we have now that were not available 20 years ago when people were first thinking about this. So obviously organoid approaches offer one way to do it. So I think it's a bit early days, but I think it's something that's worth thinking about, having these different orthogonal approaches, having both the sequencing and then also developing functional technologies that can provide ancillary information or might even provide information that cannot be determined from sequencing.
Oh, look, absolutely. I wasn't trying to say it's not possible. I guess it's just, where is the fundamental next step going to really engage with the timeline that we know is needed. I can't actually see any other hands up. Can I ask you one other general question then?
Oh, sure.
So you described an amazing repository of cell line models. Do you have a governance process by which other members of the community can collaborate with you to have shared research applied to those resources, or how do you manage them?
That's another really a good and topical question. So it's something that I think we're just learning now in terms of what is the best way to collaborate and share this information and share the models. I think there are definitely challenges in sharing both, because obviously we're MTA restricted with most of our models, so that because the models are coming from 30, 40 different sites, that essentially means we have to go back and think of a process that allows us to be able to share these models. But at the same time, I'll say that we, by providing even the information of what models are out there, that already gives people the information to at least know what's available, even if it means they can't get it directly from us at the moment. We can direct them to the right source. We can tell them which model is working, provide some details about how to get the line to grow, and then also working on mechanisms of data sharing. But that's it's something actually we didn't anticipate when we first started our program, all the governance and restrictions, but yeah, definitely an issue to work out in the next year or so.
Always a challenge. Peter Fuller has his hand up. Peter.
So, Ron, I want to ask you about bleach. I mean, clearly it's got presidential endorsement in another context. And to be less trivial, but maybe still trivial, a lot of the early cancer therapeutics were based very heavily on a therapeutic window, that ultimately at a concentration they would be toxic to everything. The question is, in a single concentration screen or a single knocker, are you potentially missing drugs that in the context of a given cancer may have some sort of therapeutic window?
Yeah, that's a very good point, Peter. So initially, we did our drug screens using a single dose point. So it's definitely something to consider, but we used a high concentration with the idea that if we see some activity at micromolar concentration, we can actually go back and do a more granular dose response point. That's exactly what we're doing now, because we've done about 150 screens with the high dose point. And actually what we found was that the majority of drugs don't hit at all. So most of them are non-effective. So we in order to not continue to just waste our effort screening across 2,500 drugs, of which half don't screen, we've now actually been able to customize our library across about 800 drugs that at least show some activity in a % of cell lines, and then use a 7-point dose curves for that. So that's a really good point. And with the CRISPR, the caveat there, it's a knockout screen. So again, you gain a lot from CRISPR in terms of selectivity compared to RNAi, but what you lose is maybe the connection that RNAi has to drug effects in the sense that you're not inhibiting the gene, you're actually completely knocking it out. So, I mean, there's advantages and disadvantages, obviously, to both techniques, but I think in the case of CRISPR, the advantages outweigh, the disadvantages currently.
Okay, look, given that it's after the hour, but almost everyone has hung on to this point, we really should wrap up. So, Ron, thank you very much for presenting the first seminar for the series. Fantastic start, inspirational for many of our work, I think, to try and think about the complexity of what lies behind the biology of what we're looking at. So thank you very much for presenting.